Thank you very much. So I'd like to go ahead and ask Ravi, as well as our two additional panelists, to join us up here. We had a last moment change. Donna Mendrick unfortunately wasn't able to join us today. And so Nikisa is agreed to step in and participate once more as an FDA, provide an FDA perspective from Pharm Talks. And then we also have joining us is Jonathan Jackson from Pfizer, where he is a senior principal scientist within their Global Discovery Investigative Talks group. Okay, so yeah, again, thank you all of you for joining us up here today. And so, to kind of lead off here. Qiang, within your results, looking at the various enzymes and activity compared to concentration, is that indicating to you that there's an unfolding issue in the serum that's being, or in the matrix that's being used, or something else going on in those studies?
This is a really very good question, because we had a lot of internal discussions about why the activity levels are not stable. So this is apparently unique only to ALT, but not AST. I believe the major factor is that the environment and the concentration, because our findings are actually in line with the literature reports in early stages when people wanted to isolate and purify ALT. It was noted that these enzymes are only stable under certain conditions, like when you add something to protect its activity, like a glycerol. You need a high concentration of glycerol to maintain the activity. So I believe it's mainly the matrix effect.
So then, would you propose in the future using an ELISA or a protein concentration measurement for ALT, or would you think that a different biomarker would be a more suitable measurement of hepatic injury?
I think ALT is definitely the way to go, but honestly, we only tested one system, so I don't know if our findings are applicable to other systems. Probably with all the different systems, you need to test your own matrix to make sure if it works, we just use activity. If it doesn't work, we may switch to alternative measurements, way of measurement, I mean.
Thanks. And to the others on the panel, have you had similar experiences with other platforms in terms of measuring ALT activity and seeing it significantly decrease over time?
Yes, we saw that in our some of our microphysiological systems when we used to develop in during the academic settings. I think internal things sometimes you can disclose, but as Jing mentioned, I think we don't know at this time even ALT is the best marker to measure in the complex in vitro models. I think it's one of the best, but we need to develop like a multi-parametric assay panels to profile a group of markers, because now the technology advances. For example, O-link kind of technologies where you can use the microsample, just 1 or 2 microliters. You can measure the hundreds of analytes. So if you doing the qualification, at least we need to think about and developing those kind of panels, and we need to create like a benchmark, a panel. I think then we can implement those things while testing our drugs. So I don't think so. Just going to one is not a great idea. So,
Jonathan, do you have an additional comment?
Yeah, I actually had a question for Qiang. So great presentation. But I was curious to understand. I mean, I understand why you make the rationale for bilirubin because it's technically not in the system, right? But I'm curious as to why you wouldn't consider the option of exogenous application of it as a functional tool, just like it's utilized in vivo? I mean,
Yeah, that's a valid question, a good question. The simple answer is because we are lazy. I mean, well, I mean, I actually looked very hard into the literature. I believe there is only one paper showing that it is possible to measure bilirubin-related metabolites. It is indeed possible, but there's only one paper. I don't know how reproducible it is, and I think it takes lots of efforts to set up the message in the system.
Yeah, measuring bilirubin can be challenging, especially bilirubin glucuronide. I would actually pose that an alternative would be coproprophins. They're actually utilized in some ADME type applications to understanding if you have OP1 OP transporter inhibition as well as efflux issues. So that's also a potential route where we could go to actually introduce coproprophins, and that would actually give us quite a bit of information regarding uptake and efflux.
Interesting. Nikisa, did you have anything else you'd like to add?
No, I don't have anything to add because I'm not doing the lab work. We kind of, Qiang is doing it, but I think what he highlights in his work is the importance of having to look at these types of parameters. And, as he said, this is with a single platform. We would have to see with, different types of platforms. And so that kind of illustrates that in any type of sort of validation or consensus that we would want to reach, we really have to evaluate parameters from different perspectives, not just, enzyme activity, but protein level and in multiple systems and multiple conditions in order to be able to generalize the conclusions, because otherwise it becomes really not applicable from a regulatory perspective where you're really trying to generalize the results. And I also agree that trying to find something for bilirubin would be, very good, because I think that would be, I think right now it's not something we can measure, and it would be nice to be able to have that kind of assessment.
Also, when you perform that kind of validation, it's a good idea to compare the single donor cell type across the different platforms. So, because donor to donor variation, all those things you need to consider. So, we need to consult.
Okay, yeah, thanks. And definitely, as we're thinking about what types of study designs are appropriate to test the context of use, I think that's a very good point to bring up. Let's go out to the audience. I think we've got a few questions here right now.
Can you hear me?
Okay. Yes.
Yeah. Maybe this question is to Ravi. This is in connection to my earlier question about the validation. What we are looking for biomarkers. As he mentioned that one biomarker is tricky to believe in and how it is performing considering technical challenges. But Ravi, when you mention about the omics, looking at the multiple aspects, so do we have a bridging between that? So it is, as per my understanding, it may not be possible to have a biomarker, what you see in the clinical outcome, the same thing can be connected to the CIVMs. So when we are proposing to use the omics, how do we bridge the gap between these two?
So that's a good question. As mentioned earlier, if you develop an multiparametric biomarker panel and you started using that platform in assessing those biomarkers in the CIVM models, and what kind of drug you are going to test and what kind of issues it's in the clinic, right? And we need to identify those patient population and for those studies who performed it, and try to get those samples and testing against the panel what you developed. And because as I mentioned, this kind of Olink kind of a platform, just 1 or 2 microliter, you can profile in hundreds of analytes. So then you can try to bridge the gap in terms of a biomarker and also the drug response, all the answers you will get it. That's how, but you need to have access to those biobank and all, those are very critical. So.
Thanks, Ravi. Anyone else have a response?
I was just going to say that, I mean, in my opinion, I think it depends also on the context of use, whether you're trying to predict something or you're trying to kind of, describe a mechanism that also, so it's assessing risk versus predictivity are two aspects, as you may not need to do that if you kind of have an idea of what you're trying to show and you want to rule it out, for example.
Thanks, Nikki. So that's an excellent point where if we're looking at things retrospectively where we already saw a signal in the clinic, we can kind of really narrow down the specific outcomes or endpoints that we want to measure in the model. Whereas if we're looking at predictive, we may need to have a cast a wider net. Thanks. Eric?
I'd like to better understand how the addition of some immune cell types like stellate cells or Kupffer cells will add to the value and what we learn from these in vitro models. Because it seems to me that all types of liver injury, whether it's drug related or not, and then all of the different pathways of drug injury to the liver would stimulate these cells in some fashion. And so are they always activated regardless of the injury, and what value do they add?
Qiang, I think, maybe you might, because I think that you have some experience with that.
Okay, yeah. So in our study, we are co-culturing primary hepatocyte, human hepatocyte, with endothelial cells, stellate cells, and Kupffer cells. These three non-parenchymal cells, they are mixture. They are cultured in different channels. We didn't do the test, but according to numerous reports, co-culturing these cells would promote the growth of each cell type. So this will enable the hepatocyte to grow longer in the systems. And also, this is according to literature reports. So some drugs can hit only, let's say, Kupffer cells, but doesn't really affect the hepatocyte functions. So this kind of complex systems can help answer these types of questions.
Thank you. Yeah, so it helps with the viability of the system, but may not add so much insight into the specific mechanisms or pathways of injury?
Indeed, I think according to some report again, so some drugs can activate Kupffer cells. So once they are activated, or stellate cells, so when these cells are activated, numerous cytokines excretion are increased. I think that may provide some mechanistic insight.
Thank you. And can I ask one more question?
Unless someone else has other responses to your first question.
Yeah, I was just going to say that I think by virtue of increasing the longevity of the cells, I think you are probably going to be able to look at additional endpoints of toxicity just because of the fact that you're creating an environment where the cells can actually last longer and sort of be in a more, in a type of architecture which is more similar to the in vivo situation, the likelihood of being able to, but that's something that we will have to evaluate with actual data. And I think that, one of the things that we are interested in trying to understand is if in multiple species that can also happen or not, because some of the challenges with getting some other species cells to grow in this type of 3D situations because of the lack of available Kupffer cells. And so that may make the sort of, rat hepatocytes not last as long. And so I think that being able to get them to grow longer then will allow you to be able to actually measure additional types of endpoints and, additional mechanisms by which toxicity might be occurring.
I would just add on to the fact that, there's literature evidence in the animals that demonstrate that, these other cell types do play a role in exacerbating damage that occurs through drug exposure. I think when it comes to humans, though, while I'm sure that translates, I'm sure a lot of this stuff, a lot of these interactions between the cells will actually become a little bit clearer to us as these models evolve.
Yeah, go ahead, Eric.
Okay. Thanks for those answers. And my last question is maybe a bit provocative, maybe a bit naive, or perhaps a combination of both. And that is, with so many of drug-induced liver injury being rare and unpredictable, on the order of maybe 1 in hundreds of cases or thousands of cases, why should it be that with these in vitro systems, it's not equally as rare and unpredictable and only happening infrequently and therefore not seeing the results that we would want to see?
I was just going to say, you're absolutely right. I mean, I think it's going to be very difficult to be able to predict an idiosyncratic unless, drug DILI, unless you can have enough types of cells with the kind of, phenotypes that are necessary to be able to represent an entire population, which is unlikely to happen. And so, it's not all just the idiosyncratic types of there's indirect type of DILI that has an immune component to it. And so I think that any type of improvement in the prediction or sort of description of DILI would probably be beneficial at this point. And so I think that the ultimate goal would be to try and be able to get to the idiosyncratic responses, but I don't know that we are likely to be able to get there just based on CIVM. I suspect that there's going to need to be additional tools that are going to be used in addition to these complex in vitro systems, which may include artificial intelligence and other tools that can take humongous amounts of data to try and narrow them down into something that makes more sense. So I think it might be too much to expect from just the CIVMs, but it may be part of trying to get there. But I don't know, maybe people have better ideas.
Eric, I think that's a fantastic question. I think that kind of gets directly at the heart of the sheer challenge of what we're trying to do here. I think that that question really demonstrates the fact that these models have to be complex in order to actually become a predictive model, and you can only do that by recapitulating the complexity of the organ, which includes all the cell types. It includes the 2-compartment model. But also at the same time, we also need to bring in genetic diversity. How are we going to do that? We also need to think about phenotypic diversity as well, because there are things through behavioral occurrences or whatever that we get that phenotypic diversity. So it probably will take a multi-tiered type of strategy. The genetic diversity is something that maybe we can use donor pools. Maybe we can think about these donor pools. Maybe they could be combined in using ethnicities. Because we know that polymorphisms tend to be enriched in certain ethnicities, certain polymorphisms. So, that's a strategy. It may be not the strategy, but, we definitely need to overcome that challenge.
Thanks. Anyone else on that question?
So one more thing is that, yes, when all the initiatives started a couple of years back, everybody thought the complex in vitro models, especially organ-on-chip models, they thought we are going to solve all the issues. But day by day, when we started generating data, when I look into data, I don't think so it's going to solve all the issues. Probably, as in the previous speaker mentioning, it's a case by case actually. So one step at a time you need to take, and based on that patient population, we need to understand, then we need to move forward with that. So, and also during all the interpretation and integration, we need to consider all the aspects from the preclinical to the clinic, all those things. And as like I mentioned, AI tools may be beneficial in that direction. We are seeing some of the things, and some of the companies are developing those tools, and they're showcasing some of the data in the case of NASH patients. I saw those things with live demonstration few weeks back. I think there is a potential associated with it. Yeah, it's a case by case. So
yeah, can I add a little bit? So I noticed a report. So it's an interesting study. So basically, a group of patients were treated with some tyrosine kinase inhibitors, and of course, some patients developed liver injury. Some patients just are okay. So then the blood of these patients were collected, and they were reversed back to induced pluripotent stem cells, and then these cells were differentiated to hepatocyte, and then these cells were treated under in vivo conditions using the same drug, and they saw the different toxicity profiles, and these profiles correlated very well with the in vivo responses. But this is a pretty recent study. I'm not sure if that can help.
Thanks. Did you have a comment, David?
Yeah, I had a thought on the last.
Okay.
Are we out of time or?
No,
something?
we've got time still. There's one other question. That
Oh, no.
But if you want to continue on the current question.
Oh yeah, I was going to give the opposite response to that question that was posed. of what I mean, one approach is you try and make the systems super complicated, and you incorporate what you were saying, Jonathan, you use, I don't know, AI, or the other approach, which is what's done with like DILIsym, the quantitative systems toxicology model, is you have some very simple targeted in vitro assays at a very specific mechanism, and then you combine it together in a computational model. But it gets at, you can try and create a super complicated system that has all possible mechanisms, or you break down the understanding of what's like the most likely reasons you can create an injury. I mean, so, and there's advantages and disadvantages to both, but it's something to think about, like, and yeah, I mean, DILI's complex. We heard about that from different talks this morning, but you know, where's the biggest sort of return on investment of assays, and something to think about.
Yeah, no, I think it's definitely, do you throw it all together and see how they interact, or play with each component and see which ones increase and decrease depending upon injury? It's definitely a balance and discussion to be had.
Yeah, I mean, unfortunately, I mean, for the last 2 decades, we've tried that fit for purpose, right? Where we've broken it down to B-cell inhibition and mitochondrial dysfunction and oxidative stress, and then we're adding or summing all of these, you know, parameters up to make a prediction. We haven't been very successful doing it that way.
And Kevin, I think you've got a question.
Yeah, well, I do. So I just have a quick question based on my previous work as a drug developer. It occurred to me today, based on the discussion of ALT as a biomarker and the discussion on Hy's law, how the diagnosis of DILI is based primarily on, or in a large degree, on the activity of ALT, which made me think of, do we ever consider the possibility of a drug candidate having the off-target effect of potentially inhibiting ALT? So that would suggest that ALT activity would be reduced, whereas a protein level could actually be quite high. So there could be a disconnect between activity and protein levels. So just wondering if any of you guys have ever heard of something like that.
Are you saying that the drug itself is actually causing the activity to go down, but the protein? I mean,
if the drug inhibited ALT.
Yeah, I don't know if, you know, it would have to run proper control for that one, because I don't think that you added exogenously afterwards just the drug to see whether the activity would be affected. I mean, I suppose it's possible, but this was without. I mean, you saw it also without the drug too, right?
Yeah. This is a really good question. And like Natasha said, we needed to include the correct control. The drug itself, drug containing medium, is a needed control, and that's something we always do. Indeed, that we indeed notice that some drugs at a very high concentration can inhibit ALT, and particularly LDH. LDH seems more sensitive to inhibition by drugs.
Next question.
So my question is actually related. Oftentimes, reactor metabolites form covalent bonds with proteins, nucleic acids, lipids. So that might be one way, but we actually had a situation where we were able to backtrack that and figure out where the problem was. So one of the suggestions are protease inhibitors. And the other thing is just staying away from enzymatic assays, although you're going to run the same problem with ATP. So you've got to run multiple ones and use a ratio of the sum to get to the bottom of the problem. That's what we figured out worked for us.
That's definitely an interesting
Thank
consideration.
you for sharing.
Yeah, I think that these are important things to consider when developing best practices when, in order to try and generalize some of these models. And that's the kind of data that's really needed, and people need to share their experiences so that we can then consider these. I think people look towards the regulators to provide guidance about these types of methods, and it's important for us to know what some of the challenges are in order to be able to figure out how to provide the guidance that's needed in terms of what are the best practices for generalizing these types of tools for everyone then to use under the correct context of use.
Yeah, definitely. I mean, this is where it comes in, where negative results are important to publish. And forums like this are a great place to bring up results like that, because we're doing this as a community and not any one individual. I think we've got a question from the online audience.
Along the line, I think the data sharing is also critical, right? Because if they already have a data, and if they're able to produce somewhere around in the public database, they're able to deposit, then we can use the data so that we can accelerate this qualification for the qualification of the CIVM model. So,
yes, definitely.
So I've got an online question here to Ravi's talk, and going back to Kevin's from before. It's easy to identify a small set of compounds and test positive and negative controls, but as you add more compounds, it becomes more complex and more cost prohibitive. Where do you see the sweet spot in the designing an experiment to have confidence in the model without being too prohibitively expensive or onerous on the researchers?
that's a very good question, actually. So, when we think about toolkits, right, I think the one, I think Kevin also mentioned where I also showed that one, I think they showed around 20 compounds with the different Bailey mechanisms. Yes, there are some limitations there in those compound set. Then the question is, when we are characterizing these models or when we are performing fit for purpose validation, just a compound, not only compounds are important, but you need to think about the other parameters too, right? You talk about the cells, cell types, number of cells, endpoints. So we are creating so much complexity in every step of qualification. That's what we're going to face. So I think selecting, few positive controls, few negative controls. I think the drugs, those are pulled out of the market. They have a specific reason, or some of those category. I think some of the derivatives of that. I think you can, if you make a small list, because as I mentioned, if you're going to test multiple donors or multiple cell types, I'm pretty sure there's going to be huge set of experiment you need to perform. So at least 20 to 30 is a minimum number, I would think. But different people have different opinions.
If I might step in for just a moment, I think there's two components to that, or two different areas where you want to answer that question. One is going to be around the model validation and characterization early on, so that we have confidence in what that model is doing. But then the other is going to be when you are looking at a qualification and answering the specific context of use and testing that. And those are going to have two very different numbers in every case. I think with a context of use, if you have a very narrow context of use, there won't be nearly as many compounds that you would want to test. It'd most likely be positives and negatives, and a few of those, and probably have a higher N of donors over number of compounds.
Couldn't agree more with Nick, but I'll also say that we should reach out to our biostatisticians.
All right, are there Other questions right now? Well, if not, I think we might be able to end this session a few minutes early today. Any departing comments from our panelists?
Last thing I would say is we should think about adding things to the model. Like, for instance, we were talking about the bilirubin. We should also consider bile acids as well. This is something that's not static, it's dynamic. So in order for us to really truly understand cholestatic liability, we should consider adding bile acids exogenously to these models, and then testing both in low and high bile acid concentrations, as this is an added complexity that isn't really that difficult to do.
I will say that's one thing that we're doing now, so stay tuned for the results of what we have going on.
All right, thank you.
So are you trying to measure bilirubin, or what are you specific? What are you talking about?
We are using both bilirubin and bile acids. We're trying to do it basically forward and backwards, at least to see what the results will be, to see if it's even possible, because like we said, there really isn't that much literature out there when it comes to that. So we're trying to hopefully put some more out there so that we can progress and move forward with these students. So.
Yeah, I'm really looking forward to it. Thank you.
Thanks. And I just want to add one more piece in terms of this session and what I would like everyone to be thinking about tonight and as we go into tomorrow is when we're given or approaching and considering a specific context of use. Once you've gone through your model validation, what is the appropriate test? What's the appropriate study? What are the appropriate endpoints for testing your context of use in order to demonstrate its validity? And that is what I would really like everyone to think about tonight coming out of this session, in addition to the biomarkers and endpoints and different components, but also stepping back and thinking in the big picture of a qualification. So thank you everyone for your participation here, and I think we have a small closing by Graham Marsh.
So just a short thank you to everybody for your participation today. I think this was a great discussion, and I'm excited to see such cross-talk between the stakeholders in this room today. For tomorrow, we will have breakouts, and if you've not chosen a breakout session yet, please do. I don't have the QR code up here. That was an oversight, but I believe an email went around. If you have any questions about the breakouts, feel free to come up to me after the end of the day here. But really, thank you, and we'll see most of you tomorrow. Oh, Danny, were you? Yes. So I don't remember the exact wording off the top of my head, but it was using CIVM to de-risk a drug with a known toxicity in class, right? And the thought there was that if you have a drug with a known toxicity, a known propensity for DILI, if you have a similar structured drug, can you put that into a CIVM model, show less toxicity than the other in-class compound, and bring that forward for a regulatory decision? That was the thought. I believe there's one example of organoid data being used in that way in regulatory decision making. So that was one of our potentially lower regulatory bar examples that we were thinking about. Okay, so thank you everybody, and appreciate your input today. It's been great.